In the next section, we're going to talk about the growth of bacteria, the way they grow, and the speed at which they grow, and basically their characteristic growth pattern. Bacteria divide through an asexual process. Bacteria don't have sex, so it's an asexual process known as binary fission. And basically the way this works is that we start with a single cell, and when the cell is ready to divide, the first thing it's going to do, enlarge. So it's going to become wider, and it's also going to double a lot of its parts. For example, these little blue dots are the ribosomes, so it'll double its ribosomes. It'll double its cell wall, and then in the next step, it's going to double its genetic material. Remember, bacteria have one circular chromosome, so that's going to make an exact copy of its circular chromosome. And then a wall is going to start to form down the center of the cell, and this wall is called the septum. And that's where the cell is going to split when it's ready to divide into two. Now, in this picture, it's almost complete, and then finally the cells break apart, and we really go just from one cell to two. And then those two cells will divide, and we'll have 4. And then those 4 will divide, we'll have 8. So it just keeps going. And as you can imagine, for most bacteria, this process of binary fission happens pretty quickly. The time that it takes for one cell to divide into 2, time it takes for it to double in number, we call this the generation time. And another name for it, aka, is doubling time. Because each time a cell divides, each time one cell divides, it doubles in number. Let me give you some examples. E. coli's doubling time is about 20 minutes at its optimum temperature. Okay, so every 20 minutes, one cell divides into 2. Most common human pathogens, the doubling time is going to be about like 20 to 40 minutes. There are some weird exceptions. Okay, so for example, Mycobacterium. Mycobacterium are those acid-fast bacteria, remember, with the waxy cell walls? One example is Mycobacterium tuberculosis. So like the name says, this causes TB, tuberculosis. Its doubling time is not even measured in minutes. It's actually about 12 hours. So it takes about 12 hours for Mycobacterium tuberculosis to double. Another one, Mycobacterium leprae, which causes leprosy. This one is so slow growing that it takes about 12 days for one cell to divide into 2. And that's why leprosy, it takes so many years to fully develop. Another example, Treponema pallidum. This causes syphilis. And syphilis is also a really slowly developing disease. Treponema has a doubling time about 1 day. So it takes about a day for the cells to double in number. But remember, for the most part, pathogens are going to be about 20 to 40 minutes to divide. And the process is going to happen so quickly that it's actually exponential growth. So binary fission is actually a form of exponential growth. To calculate the number of cells in a population, we have this very simple formula. This is basic algebra. So, for example, NF, this is the total number of cells in the population at a given time. Ni is your starting number of cells. And then the N is the generation time. Okay, so let's take an example. Let's say that you are at a picnic and you're eating some chicken. And initially, the starting number of cells in your chicken is 10. So you just got to the picnic, you're eating some chicken, there's 10 bacterial cells in that chicken. That's not a lot. You'll probably be fine if you eat it. Now let's say that the generation time for the cells that are in your chicken is 20 minutes, like E. coli. And let's say that you leave your chicken sitting out in the sun for 4 hours. So that means every hour the cells are going to divide 3 times every hour, because there's 60 minutes in an hour, and they divide every 20. So the cells are going to divide 3 times per hour. So after 4 hours, the cells are going to divide 12 times. So your N, this N right here, is 12. Your formula is now NF equals 10, the number of cells originally in the population, times 2 to the 12th. 10 times 2 to the 12th is 40,960 cells. So after your chicken has sat out for only 4 hours, it has gone from 10 to about 41,000 cells. And as you can imagine, if you ingested 41,000 cells, you're probably going to get a bad case of food poisoning. The characteristic way that bacteria grow, if we are growing them in an environment, such as maybe in the human body, or let's say in this example, in a test tube in the laboratory, they grow by this characteristic growth curve. And this growth curve is really important. There are 4 phases to the growth curve. So right here, this graph, this bottom axis is time. Usually we measure it in hours. And then on this axis, this is essentially the number of cells, the amount of cells in the population. The first phase, so phase 1, is the lag, L-A-G, lag phase. All right, and that is shown right here. Okay, so from the time the bacteria have entered the new environment till about right here. This is the lag phase. What's happened in the lag phase? The bacteria have entered a new home. They're not used to it yet. They need to start synthesizing new cell parts. They have to synthesize enzymes to be able to replicate, to be able to divide. Okay, so they're kind of just waking up right now. So you don't see a lot of growth. It's kind of stagnant. Then they get into phase 2. And phase 2 is called the exponential phase. And another word for it is logarithmic or log phase. So the log phase, as you can see here, starts at the end of lag and goes to about here. During this phase, cells grow at their maximum rate. Okay, cells grow at max rate. This is where that generation time is calculated. So generation time calculated, and now the cells are growing rapidly. They're happy. They're in this new home that has a lot of nutrients. They have a lot of space to grow, and this is where you're going to see the most cell growth. A couple other things. This is where antibiotics are going to be most effective. Antibiotics usually require living, actively growing cells. So this is where the cells are living and active. Antibiotics are very effective here. Here in the middle of the exponential phase, so in the middle, bacteria are producing what's called primary metabolites. Primary metabolites are essentially the natural metabolic products of the cell. So natural products like maybe alcohol, ethanol. Okay, so when we're producing alcohol, for example, in industry or any kind of industrial product, it's going to be made in the middle here. At the end of exponential phase, the bacteria are going to produce secondary metabolites. At the end here, the bacteria are starting to get stressed because they're starting to run out of food. There's so many cells that have grown in this tube that they're all kind of squished together. They're running out of nutrients. There's too many in the population. They're just running out of room. Okay, so they're trying to kill their competitors by producing these secondary metabolites. For example, antibiotics are produced at this stage. So some bacteria actually produce antibiotics in a form of antagonism to try to kill their competitors. Another thing that's going to happen right here is that's where if the bacteria is going to make endospores will start to form. Okay, now we're going into stage 3. Stage 3 is stationary phase. All right, right here. It starts at the end of exponential and it ends here. During stationary phase, as you can see, the number of cells evens out. There's an equal number of new cells, as there are dying. So anytime one cell dies, a new cell is born. During the stationary phase, there's not a lot of nutrients left in this test tube, so the new bacteria are essentially eating the old dead ones for food. And the fourth stage is the death phase. And as you can tell by the name, bacteria have totally run out of nutrients. There's not any food left. There's all crammed up in this little test tube. There's no new nutrients. And there's a lot of waste that have accumulated. And so the number of cells is going to decline. Those cells are going to die. If you want to review all of the stages, this slide explains what I just told you. So this slide is basically showing the number of viable, that means living cells. And if we want to essentially determine the number of living cells, we will do what's called a viable plate count. Because on your petri dish, on your plate, remember every colony, when there's a colony that forms, a colony came from a living cell, came from one single living cell. So for each colony, that indicates there was one cell in that sample. So for example, if you consider the regular growth curve, starting with lag, going into log, stationary, and death, your cell counts are going to look similar, okay? So at the beginning, you're not going to see any colonies in your tube because the cells are in lag phase. And then as you go up more and more, you're going to see an exponentially growing number of cells.